on this busy time of year. I am the program director. My name is Charles Cywin, and I have been here at NINDS for 8 years today. So it is a pleasure to get a chance to tell you about this program. So the program vision for this program is to combine the strengths of NIH and industry to bring NIH investigator-initiated ideas for novel small molecule drugs with strong disease assays and models, and combine that with industry expertise in terms of advisors, in terms of our subject matter expert consultants we have with extensive pharma experience, and industry standard contract services that we've already set up here within our network that we can make available to you. This was done in answer to sort of the dearth of these available resources in many small businesses and academic institutions, and the ever need for new neurotherapeutics. So the program is for, as I said, academics and small businesses. We're going to provide grant funding in terms of activities in your laboratories to do this work, as well as access to NIH contracts and consultants. The projects can enter anywhere along the preclinical pathway to move forward. We want to identify the best neuroscience projects for translation to the clinic and provide the necessary resources that are typically lacking. The goal, obviously, is to de-risk these programs that we're supporting to the point where industry will invest in them, and that's so that we don't do a bench to or a lab to bookshelf. We want to go from your lab to the bedside eventually. So we need someone who's going to be able to take it to phase 2 and 3 down the road. So we work with you on getting industry partnership down activities for licensing and making those plans throughout the program. We do this by preserving your intellectual property, and we preserve it in a number of different ways, and I'll talk about that in a moment. The BPN remains interested in neurotherapeutics across the missions of 9 participating ICs, institutes and centers at NIH. It's not all institutes and centers, so some diseases don't fit to the Blueprint Neurotherapeutics Network, things like cancer, particularly, or NIDDK or BIB, blood, heart, and lung, and other institutes. Those are not participating, and things in their mission would not be eligible to come to BPN. But most of the neuroscience is covered by these institutes that you would be going off after. If there's any questions, please contact myself or any of the science officers at any of the institutes that participate to discuss your program and for opportunities within the Blueprint Neurotherapeutics. As I said, projects enter anywhere along the preclinical paradigm from as early as I have the hit and I have some assays to do the SAR driving in vitro assays. I have plans for my in vivo program. That becoming a sort of exploratory and early hit to lead, all the way through lead optimization onto, I've already finished MedChem, I have my development candidate, and what I really need is drug manufacturing formulation work, GLP tox and beyond. So you can come in either as a discovery project or as a development project. And although I see it, I have a little error on the slide, it says CNS disorders, it's really nervous system disorders. And so we cover both peripheral and CNS. Our goal is to exit to external funding partnerships, other grants, or in some cases, obviously, this is a milestone-driven program. Some projects do not make it because of not meeting the milestones. We offer a flexibility in terms of the mix of contract access and grant support. Although most people come to the BPN because they want to access our contract suites and they don't have this, sometimes your lab will have aspects of this. You may have some medicinal chemistry effort going on, or you may have some in vitro admit, or even some PK work, but typically no one has IND-enabling tox studies, GMP, scale-up work, formulation, or clinical trials available to them, although sometimes we have clinical trials in some academic hospital settings. However, most of what we're going to do is going to be directed towards a phase 1 first-in-human healthy volunteer study, which most places don't have available to them. But we can mix it up. So let's just say, you know, you have the ability to do some in vitro admin, but not all, or you have the ability to do maybe a medicinal chemist on your team wants to be involved, but they really don't want to hire a whole bunch of chemists that you might have to lay off if you don't make the milestones. We have the access to our chemistry resources to do that. And we do that because we have already set up a set of contracts. And so our contracts for medicinal chemistry are through AMRI, Albany Molecular Research. Our PK Tox contracts are through SRI and Southern Research. We have a drug manufacturing formulation contract through MRI Global, or again, Albany Molecular Research, and with clinical trials either through Duke or PPD. In addition to that, if you're in the discovery phase and you need a relational database to store both your compound and biological data, we also have access to our CDD relational database. So what we do is, for every team, we make a custom lead discovery development team. And in that, the principal investigator is coupled with a lead consultant, and depending on where you are in your entry stage, a lot of times in early stages it's probably a DMPK or a med chem person, maybe your lead consultant. In later stages it might be a toxicologist or somebody with chem development experience on there. But what you have to absolutely come to the program with is the disease biology yourself in your labs or as part of your grant application through your consortium of co-investigators. You need to be able to do the in vitro assays to drive SAR related to disease biology. You need to be able to do the in vivo pharmacology related to disease biology. You need to be able to support all those kind of activities through the clinical planning as well through your grant mechanism. Other than that, the rest can be contracts. And if you're actually accessing our contracts, then you do not put that part of it in your budgets for your grants. So this is a thing that's sort of advantageous to certain grantees in that, and I'll talk about the SBIR program as well as the non-SBIR version in a second, and this can where in the SBIR program it can be really important to access our contracts versus doing it yourself in some cases to meet the requirements of the SBIR program. That being said, we can move on to, as I mentioned, any project can enter anywhere on the preclinical stage, and we have the two mechanisms. We have the PAR 18546, which is a UG3-UH3. This takes all comers, whether you be an academic, a small business, a foreign institution. It pretty much will take all comers for consideration under this application, but it is more restrictive in terms of the budget. So you cannot come in with anything over $500K in any of the years of the budget without asking permission 6 to 8 weeks prior to the receipt date. So our receipt date's February 11th. So if you were coming into this, you need to send me a detailed budget and a request to do that sometime in the next couple of weeks to make the February date, so that I have time to go through the process here, because we're coming up on our council round and trying to get this approved would take some effort. But if you're coming under the SBIR rules, which has, you know, $500K in total cost and $1.5 million in total cost for the phase 2, those don't require the permission to come in. Additionally, as I mentioned, there's some advantages for a small business to go under the SBIR side, which include fees. You can charge a fee on the SBIR side, which you can't do on the UG3, UH3. And importantly, from the using our contracts, you know, IND enabling in some of the drug manufacturing and formulation can be really expensive activities, and it's hard to meet the requirements of all the money or 50% of the money in phase 2 staying with your organization, 2/3 of the money staying in phase 1 with the organization, if the contracts are not considered a fee for service. And a lot of times R&D contracts won't be considered fee for service. So in this particular case, I should say that foreign applications, as I mentioned, that can come in under the UG3, UH3 mechanism, do be aware there's sort of an extra layer. Reviewers will assess whether the project presents special opportunities for furthering research programs through the use of unusual talent, resources, population, of environmental conditions that exist in the other countries and are either not readily available in the U.S. or augment existing U.S. resources. So it's an extra hurdle, not only after this review, the reviews will look at it, but there's another review at council for approval of any foreign application. That's not true of foreign components, but foreign applications. So the grant, as I mentioned, is a two-phase mechanism. It's either a UG3 or a U44 phase 1. We call this the preparatory phase in our funding opportunity. Everyone has to enter the preparatory phase. I get this question quite often. Can you come directly to the execution phase? And the answer to that question is no. It's an opportunity to really assure the funding ICs that what is claimed in the application is really there before they commit all the money right up front. And although it is milestone based, and we'll get into the milestones, this preparatory phase has really allowed us to get time to get some additional data that may de-risk the program. It also allows us to essentially start programs that wouldn't otherwise get started because you may need to conduct some additional studies to finish out a package fully. Like you might need to repeat in vivo pharmacology, or you might need to run a dog DRF and you haven't run that yet. So things we can do in the development phase to get you fully to the development phase endpoint. Likewise, the assay may be up and running, but it may not really be operational as a full high throughput to handle the number of compounds coming for an SAR program. That can be part of the first year milestones to get that done and deliver some benchmarking of some compounds. I see a lot of early stage projects in review talking about, it would be nice if we knew something about their in vitro metabolism to know what the issues are. And again, we can do that benchmarking work in that first year and get the data that we need to really make a stronger decision whether to go forward with the program fully in the second phase of the program, either the UH3 or the U44 phase 2. Typically, BPN supports 2 years of missile chemistry in phase 2 if you had the exploratory phase in there. If you're coming in already at the hit the lead phase, it's really going to be 2 years of total chemistry, medicinal chemistry. And then we really expect to be moving towards a candidate in most cases. The goal is the same in all programs. It is to get to IND-enabling studies and then on to first-in-human trials. And we have milestones that I have here, and if you call and we have a discussion, I certainly can share with a more detailed what is in these milestones exactly and what resources are available under the BPN for each stage of this. But you can see the milestones and the expectation for time and stage are listed on this slide. The maximum grant duration for any project is 5 years across the 2 phases, and we do expect to see progression through this paradigm all the way to IND enabling in phase 1. Although sometimes the phase 1 studies do happen through our contracts after the end of the 5th year, simply because it took the whole 5 years to get to IND filing and approval. As I mentioned, the key thing about this program that's sort of different from many programs you may see out there is that Blueprint is not looking to take a stake in your intellectual property. And that being said, inventorship is always determined by U.S. patent law. And as an extramural scientist, I cannot give up my patent rights. and neither can anybody else that works at NIH. But what we do, and that's why we use these subject matter experts, that's why we have the contracts the way we have them, our staff is really here to facilitate and not be directly involved in dictating your science in terms of what compounds to make and things like that which would be inventive. We are, and we do have contracts that are available from AMRI for MedChem and MRI Global for the drug manufacturing formulation that have what's called determination of exceptional circumstances. That's a fancy way of saying they will sign their IP to you that they develop under this project. So any compounds they make, the synthesis of those compounds, they will assign to you. If in the course of what they're doing, they make a general, like I used to use this as an example, they make a general chiral catalyst that they're going to use in your synthesis, they own that chiral catalyst because they invented that separate from the program. They would have to give you a fully paid up worldwide license that's transferable for that inventorship to it, but the vendor would own it. Otherwise, anything to do with your synthesis and the compounds you make are all belong to you. All of our subject matter experts will sign intellectual property agreements as well, and to, again, assign intellectual property to you. Any royalty arrangements and things like that, you guys negotiate on your own. NIH doesn't get involved in that, and so we move from there. This kind of work is definitely a team sport and requires lots of people, and we have a great network of people and a lot of information available, and I encourage you to check out our website. And on our website you will find that the staff is listed at the bottom there. If you click on those links, you go to our email address. There's information about the funding opportunities, how our virtual model works. If you click on BPN resources, you will get to all of our consultants and learn out who they are and where they're from and their background. Almost all of them were, senior folks in pharma previously and have a great deal of experience. Let's just say you're not ready for BPN based on you don't have all the information, the entry criteria. You don't have, for instance, the assay fully worked out and you need to develop the assay or run a screen. There is a program at NINDS called Ignite. It's run by Rebecca Roof, and if you happen to fall in the NINDS's mission, you can run things like assay development and then run a screen, or you can do some limited MedChem under the neurotherapeutic agent characterization and run an in vivo efficacy study to get the data you need for preliminary, strong preliminary data that you need for the BPN application, or create for that matter, which is biologics. And you can also do validation of animal models if you need to. That again is also a two-stage program grant mechanism and is an R61, R33 with a go-no-go decision point in the middle of that. So that helps you figure out if you don't have what you need to come in, we still have opportunities to give you some. And there are similar kinds of programs elsewhere in NIH. So that concludes what I wanted to say about the program specifics. I now want to just transition to some things I recommend you do, and then some things I recommend you don't do. So some things I recommend you do. Read the funding opportunity announcement carefully. There are specific requirements in the funding opportunity announcement, such as it calls for a table to say, here are my assays, here's the throughput of those assays, here's who's going to do those assays in my research plan, whether it's going to be BPN that's doing them through our resources or whether it's going to be yours. Having that really sets up a good conversation and a clear understanding from the analysis of your budget as well as from review perspective, what's on the table to review. So if you plan on doing stuff yourself that would be under our contracts otherwise, that's fair games for reviews to assess. But if you say you're going to use our contracts for MedChem or PK or Tox, review doesn't comment on those. Those are assumed to be industry standard and they move on. I highly recommend you read the proposal carefully and that you discuss it with NIH program director, myself, or one of the Home Institute's program directors as early as possible so that we can help you. If you're an SBIR, I recommend you contact our SBIR representatives for SBIR specific issues relative to qualifications and budgets and so forth. Definitely stick to the page limit. Don't stuff the application by trying to make your fonts really small or trying to make there's no line spacing. Because that will get your application withdrawn on you. Do put forth a solid scientific premise and do address rigor in your research strategy section. Do not bury the rigor discussion of, is this a well-powered, blinded, controlled study? Should be part of the research summary, research strategy, and you should discuss not only what you plan to do, but also your preliminary data and how rigorous that was. Should definitely be part of the research strategy discussion. Address any obvious elephants in the room. So if there's some obvious criticism about a chemical alert in your molecule or some off-target activity, assume the reviewers are going to see it too and are going to want you to address it. Because if you don't, they're going to assume you don't recognize this as an issue or it's not addressable. Clearly indicate what will be done as part of the grant, as I mentioned, and what's going to be done through BPN contractors. There's a call for a table to do that, but it's really important you do it, and you can do it not there and also in your budget justification. That way we're just sure of everything. Include budget sheets for all years of the grant. This is something that I have at least 1 or 2 applications every round I have to quickly go back to and say, can you give me a full set of budgets? You need, even though you're only being awarded the preparatory phase, the review to go from preparatory to the phase 2 section is an internal administrative review. All of your budget has to be approved by council, so we need to have the entire budget in the first place. So I need all 5 years of the budget if you have 5 years, 4 years, 3 years, whatever the total number of years is, all years. I've already touched on this and just making sure that you address rigor. If you haven't already registered in ERA Commons or don't have your DUNS number or all the other requirements to make an application, now is the time to start, today, to get your registrations done, because it can take 6 to 8 weeks to get all that stuff in and get everything ready to go so you can file an application. Consider putting your application in early. A lot of people put it in at just before 5 p.m. on the day it's due, and that's fine. If it's perfect, no problem doing that. But if there's any issues with the system, then it's a bit of a problem. But importantly, I tend to look at applications that are in a couple of days before, and if I see something like you missed a budget sheet or something's not in there that should be in there included, I have an opportunity to contact you and say, hey, I don't see this in your application. You might have a chance to correct it before the due date's over. If you're applying for a UG3, UH3, and as I mentioned, any of the years, 1 through 5, any of them exceed 500K, you must ask for permission to come in. And it requires going through a process here at NINDS to get approval for that, or at the IC that's going to be supporting your project because it's in their mission. Talk with your tech transfer and business development groups early and often. They need to plan for funding patents and licensing activities. That's another thing that can be a definite issue on the milestone side is we get to the end and they say, no, it says, well, who's going to pay for patents? It's got to be you guys because NIH will not pay for that. Some things not to do. Do not plan on this to be your sole funding for your lab because it's a milestone-driven program and can end abruptly for any number of reasons. And although it's rare, it does happen. And for scientific reasons, projects will fail. That's part of what drug discovery does and is about, and it's not unusual. So again, that's another thing that leveraging BPN contract can be really good for, because if you don't have to hire them, you also don't have to fire them. So therefore, our contractors can go up and down with things pretty easily because we have a number of programs with them, but it's a lot harder for you individually to do that. Do not under-resource your budget to avoid the limits. If you really, really need something, you plan on coming back to me later asking for money, it may not be there. So it's better to ask me up front what you really need and give a well-justified answer for why you really need it than it is to wait and to just avoid the entry. Do not plan on BPN contracts for disease biology or one-off in vitro ADME studies. We can't do that. We don't have, the contracts don't allow for disease biology work, and it's really doesn't make sense to put a big contract in place for a single ADME study. So if you need to do a single in vitro ADME study to shore up your data set, you will want to include that in your grant budget. Do not code the application as a clinical trial if you plan on using BPN contractors to do the clinical trial, because it's not a clinical trial under the grant, it's a clinical trial under our contract, and so therefore you don't have to worry about that. If you do, however, plan on doing a clinical trial yourself, it will be a delayed onset because you're not starting it on day one with us. The other thing I want you to at least make sure of is you're aware of key notices. Don't put something into an application that is not called for. It comes back to that statement I made about making sure you read the FOA carefully. Unfortunately, I've seen people do this. They append papers to the appendix, or they put a patent application that hasn't published in the appendix, and then the whole application is withdrawn before I ever see it because it has disallowed materials, and there's nothing I can do. If this comes in on the day of the receipt and I can't get you to fix it before the receipt expires, date expires, they'll just withdraw it, and they don't. There's no recourse in that case. We're accepting new applications, and the next due date is February 11th. A lot of people also ask me about how this is reviewed. It's reviewed through a special emphasis panel, an INDS special emphasis panel. We have a number of indications. If you look in the funding opportunity announcements, you will see each individual IC that I have in that first 9 there has a statement saying what they're interested in. And so from there, I'm ready for questions and discussions along the application process, across review process, proposals and budgets, contracts, intellectual property. I can answer anything you'd like along these things. I want to thank you for your attention today. Wish you all happy holidays, and I'm going to turn this over to Tim, who's going to give me the questions now, and I've got to do some quick little maneuvers on the WebEx so I can make that all work. So.
Just wanted to remind people the way that you're going to ask your questions is in the Q&A field. And one question that we always get is, will this presentation and the recording be made available? And yes, that is the case. We will have it available online and will be emailed out to you. Connection.
Still showing it on your screen.
All right. So the first question. And just bear with us 1 second. All right, so this person wanted to doubly confirm that real expenses with third-party blueprint contractors, for example, chemistry activities, GMP manufacturing, IND enabling tox, phase 1 trial, which would cost over $1 million, are not included in our budget.
Correct. Well over $1 million, and not in your budget. If you're using our contractors, we'll do that outside of your budget.
Next question. Can we reach out to vendors directly to help with the project/proposal planning?
I'm not sure if you're asking about our BPN vendors or whether you're asking about vendors on your own. Certainly vendors on your own, you're welcome to reach out and talk to any vendor you choose to, including the BPN vendors, and they can give you whatever help they deem they wish to help you with. I have no problem with that. You can't really reach out to our consultants because unless they're volunteering their time, we would incur costs. And likewise, our vendors, I can't authorize you to incur costs on the government behalf. So if they want to work with you separately from our government contracts, they're welcome to do that.
Next question. Can you be involved in both SBIR and blueprints?
I'm not sure what that means, but the Blueprint Neurotherapeutics Network has two tracks. It has a non-SBIR and an SBIR. They're both Blueprint Neurotherapeutics, and you can come in under the U44 and be a Blueprint SBIR eligible project. If you're asking, can you have an application under the Omnibus and under Blueprint simultaneously, the answer is sort of yes and no. Yes, you can have two projects, but they can't be overlapping. You can't have overlapping projects funded by anywhere in the federal government, and you can't have overlapping projects under review anywhere at NIH at the same time.
What is the pay line, and how many BPN grants get funded?
There is no particular pay line. We don't percentile these grants. They're scored through that special emphasis panel. We typically fund, I would say, 1 to 2 projects every receipt date, and it's a similar kind of pay line to what we see across anywhere else in NANDS or elsewhere. If you were trying to do pseudo pay lining it, I guess. But what I find is a better way of thinking about it is if I have an application that scores well and the IC is supportive of it, it's probably going to get funded.
All right, next question. Is it possible to connect with an experienced project coordinator who could help with project/proposal planning?
Only to the extent that I would do for anybody. So you can contact the program director like myself to give you some general advice about what's going on. But obviously as federal employees, there's an ethical issue of us helping to write an external person's application to our programs. So anybody in the federal government would be restricted from what they can do there.
Next question. Can you tell us again where we can find the list of approved vendors that are Blueprint specific?
So on the Blueprint Neurotherapeutics website under BPN resources, we'll list our contractors, not only the CROs, but also all of our consultants, as well as our external oversight committee are all listed on the BPN resource page. So you can see exactly who's working with us.
If we are using your tax facilities, do we specify what they want to do, or do they look at the program and decide what services are required?
Tox, I've said. If we are using our tox facilities, do we specify what they want to do, or do they look at the program and decide what service? Okay, so these programs, the LETs, I probably didn't mention, maybe I should have, definitely should have. This program is really interactive. We meet every 2 weeks on these lead discovery teams via WebEx type meetings like this, very interactive on the phone. Everything is done as a collaborative method. But the tox studies that we do are, we write the statement of work and they do what's in our statement of work. But a standard set of GLP tox studies for enabling phase 1 is regulatorily determined. So we have to do repeat dose toxicology, we have to do safety pharmacology, we have to do genotox, we have to do PERG, and that's really the paradigm. Before that, we do dose range binding studies in either rodent and dog typically, but sometimes primates are in there too. So our typical is rat and dog by default, and others can be considered.
Next question. If you are close but not fully satisfying the entry criteria, for example, more PD work needed for the development phase, can you propose to address the shortcomings in the phase 1 of the development stage project?
Yes. So if you look in the funding opportunity announcement, it clearly states that you can come in and say, I've identified the compound, but I need to do this stuff in the preparatory phase. It might be a dose range finding study, it might be a pharmacology. some sort of efficacy study, you want to get to that milestone that says, I have a therapeutic index in my efficacy in my rodent tox study. So yes, absolutely.
What are the success rates for the applications in recent years? Can you report discovery versus development?
I actually don't remember the success rate per se. It is very on par with, as I said, other applications in NINDS and elsewhere, NIH. It is, I would say, I'm not sure to say if it's more different for discovery versus development. I would say discovery probably does a little better on average, but that's just an impression. I don't have numbers for that. What I find with development projects, I will say this, from a review perspective, people that are reviewing them either really like them or really dislike them. Doesn't seem to be any middle ground on development projects. Either you have the package of data that supports it to being a development project or you don't. And if you don't have it and you're proposing it, then many times it won't fare as well. That's why I tell people sometimes not to overreach for where they really are on the spectrum. If you really still need to do some additional medchem and you actually need to do some efficacy studies, say you're coming in late discovery and this is what you need to do rather than say you're coming in as a development candidate, give yourself the ability to, if you haven't actually run it, you have backup compounds, you want to have the ability to characterize the compounds and make a decision between, the one or two compounds you might have there. But I can tell you, one of our early projects, one of our first projects in development is now in the clinic as of a couple of weeks ago. That came straight in development.
Next question. Are there funds available for early stage clinical trials?
I'm not sure what that question's asking. There are funds available if you wanted to do phase 1 yourself, but early stage clinical trials, meaning something beyond phase 1, where you're dealing with patients and efficacy, would be out of scope of BPN. There are other funding opportunity announcements available out there.
At what stage can one come in? We have conducted an in vitro screen that has identified 2 small molecule hits. We have no in vivo pharmacology yet. Is this a reasonable entry point into your program, or would you recommend another grant mechanism?
So it's hard to judge that with this limited information, and so I'll have to say it depends. Depends on how strong a story you can make about the scientific premise based on the data you have available to you. Having in vivo pharmacology is not an absolute requirement, and I have seen successful projects without in vivo pharmacology come in and do okay. But they had enough data in the literature to make a compelling case to the reviewers that this should work and therefore scored well. If you think you need additional data, a project could also go into something like the IGNITE program that would do you could do the additional medchem and do the in vivo pharmacology and get the preliminary data you need to have a stronger application.
Next question. If we got scored but don't have the reviewers' comments yet, should we plan for the next cycle but not the upcoming one?
That's really, well, I would say this. You will not be able to put in the application if you do not have a summary statement back, because the rule I mentioned of you can't have 2 under review at the same time. So as soon as the summary statement is back, it's no longer under review and you can put it in again. If you wish to come in for the next cycle, you should contact the scientific review officer. If this is a BPN grant, you would contact Joel Sadoff. If it was another grant application somewhere, you contact whoever's listed running that study section and say, hey, I really need my summary statement because I'm planning on applying to the next round. And he should be able to get that to you, or at least put it to the top of his list so that he'll get it to you sooner rather than later. So can BPN perform a limited phase 2 trial on a rare GNS disorder as part of the grant? And the answer is no. It's specifically excluded from the grant, anything beyond phase 1. There are things like the exploratory clinical trials, FOA, that's part of the Division of Clinical Research here at NINDS that can help. We are proposing a project that is ready for phase 1 evaluation in man. If it is strictly meaning you've already completed all your toxicology, the IND is filed and is ready for phase 1 in man, the answer is no. You should go to what I just mentioned, the exploratory clinical trials, funding opportunities, or something like that at another institute, if it's not an NINDS project. If, in fact, by ready for phase 1 meaning you need to go do the GLP tox studies and get all that formulation and be ready for phase 1, you know, manufacture the material for GMP, all of that needs to be done prior to running phase 1, then it is appropriate for this. So again, devil's in the details of exactly what you have. Most people are not, when they say this, I'm going to assume you probably are going to be acceptable to come in because you still need additional activities to be fully ready for phase 1 with materials in hand.
Okay. How much detail about BPN vendors' statement of work, example, GLP tox or phase 1 trial design, should or need not go into the proposal?
You do not, if you're using BPN vendors, you do not need to spend much effort at all in your research strategy. If it was, I would recommend saying something on the order of, our plan is to use BPN vendors for our GLP tox to run the standard battery of tests that include repeat dose toxicology, safety pharmacology, genotox, and HERG, and that would be the end of my sentence. That way the reviewers understand that you understand what's required and that it's going to be done.
Next question. I looked up and it appears that IGNITE is specific to NINDS. Is that correct? Does NIMH have funding opportunities similar to what IGNITE funds?
I'm not 100% positive of what NIMH has for similar kind of funding opportunities for that, but I would recommend highly that you contact Enrique Michelati, who's the contact from NIMH on this grant, and he will certainly be able to give you more information for that.
And to follow up on that, IGNITE, we did have an IGNITE webinar 2 weeks ago. If you are interested in that recording or the presentation, please reach out to us and we can send that your way. Is Blueprint unique in the off-budget work provided by BPN vendors?
Unique. Well, the program is pretty unique at NIH, so I'm not saying it's not 100% unique because I've been there 8 years, and at this point there are other ICs that are taking some of our successes and trying to mimic them in their programs. So I've seen this work done for NIAID has a suite of contracts, NCATS has a suite of contracts, BIB or NHLBI has a suite of contracts, NCI predated me and has a whole infrastructure related to both small molecules and biologics associated with cancer research as well. CREATE does not at this time offer access to contractors other than consultants for a few very focused areas. Again, I would talk to Chris Boshaw about that particular question, who's the program officer for the CREATE Bio program. He can give you more information.
All right, we are at the end of the line. We do have a couple more minutes, so if you'd like to ask a question, you can jump right in. We're going to give it, I guess, around 1 minute to give people a chance to ask any questions they might have. And like I said, we will be sending this out to everybody that has registered. If you are just calling in to the webinar and didn't actually register, please reach out to us so we can make sure that you get those materials.
Yes, we have a very good listserv that will give you updates on our opportunities, not only for this program, for other programs, NINDS and elsewhere have to offer.
And I would also strongly suggest you follow the Twitter account that's at NINDS Translate. That's at NINDS Translate. On that account, you'll see all of our funding opportunities promoted, as well as more webinars, or when program officials will be joining, going to conferences. All right. So if we are uncertain about the stage of the process for our project, I assume we could just contact you and have a chat, correct?
Absolutely. I always recommend people contact me. Tim manages my calendar, and I try to get on the calls when Tim assigns them to my calendar religiously. And so the best thing to do is, if you want to set up a call, is contact me or Tim and ask to be on to get a call set up with me. I try to get them scheduled as quickly as possible right now, and Tim does a great job of getting them in, within a couple of days of your request in most cases.
All right, we have another question. So if I have all my IND enabling work done, but do not have a finalized human assay method as of the application time, is this application method appropriate?
I'm not sure what they're asking exactly there. If all your IND enabling work is done, then you're probably beyond us, and you probably want to go to a clinical trial funding opportunity announcement. I assume what you're asking is, bioanalytical method developed for human PK studies, and that was typically done by the clinical CRO.
Doug, if you'd like to follow up, get a little bit more clarification, you're more than welcome to email us. And I think with that, we're going to come to the end of this webinar. Thank you all for joining. If you all have any suggestions on how we can make these webinars better, please let us know. If you have any suggestions for new topics that you'd like us to cover, please let us know. I hope everyone has a happy holidays, and thanks again.
Yes, thank you very much. I really appreciate your interest and look forward to